following best describes the two divisions that occur in meiosis? So look, the first division, which is meiosis 1, takes a diploid and makes it into two haploids. And then meiosis 2 takes those haploids and simply makes them into more haploids. So A says the first cell division, which is meiosis 1, results in two diploid cells. No, it results in two haploid cells, because N is for haploid and 2N is for diploid. So A is going to be off the table. We only use the term homologous chromosomes to talk about meiosis 1. So in meiosis 1, we do mention the term homologous chromosomes. In meiosis 2, we use the term sister chromatids. So let's see if that's what they did in part B. Homologous chromosomes separate in the first division, true, and paired chromatids separate in second division, true. So it's choice B. Okay, question 2. Here we have two polymers. Polymer means multiple monomers that have been put together. And we can see how they are both made of the same subunit, which is probably glucose. But in polymer X, all the CH2OH are on top and all the OHs are on the bottom. But in polymer Y, you switch. So one time CH2OH is on the top, one time OH is on top. One time CH2OH is on top, one time OH is on top. So they keep switching. So this is called different orientation. So the polymers are made of the same thing, right? The same subunit, but the orientation is basically different. And of course, if you have different orientation, you have different structure. And if you have different structure, you end up with a different function. Okay. Now, both polymers get broken down by hydrolysis reaction, both of them, regardless of their orientation. So it's not going to be A. The order of the monomers is important to the function of the polymer Y, but unimportant to the function of polymer X. Very wrong. Order of the monomers is important to both polymers, not just one of them. The polymers differ in the type of the atom they contain. Not true at all. They both contain the same atoms. They both contain carbon, hydrogen, and oxygen. So C is off. The orientation of the monomers relative to each other differs in the polymers. True. Question 3. Here we see a bar graph talking about enzyme activity as the y-axis. And we have multiple ions on the x-axis. Some enzymes require the presence of certain positively charged ions to be fully functional. To determine whether a newly discovered enzyme requires the presence of positive ions, researchers determine the activity of the enzyme in solutions containing different ions. Which ion do you think here gave us the most enzyme activity? Mn, exactly. So Mn here gave us the most enzymatic activity. Now look at A. Mn increases enzyme activity, whereas the other ions do not alter enzyme activity. If you want to assess whether the other ions alter the enzyme activity or not, you must always compare them to the control. The control is going to be no ion. And guess what? All the ions have a higher enzyme activity compared to no ion. But just that Mn has the most one. So A is off. Mn2 plus increases enzyme activity, whereas the other ions lower it. بالعكس. Again, compared to the control, all the ions increase the enzyme activity, but Mn being the highest. Mn2 plus is necessary for the enzyme catalyzed reaction to take place. I mean, maybe, right? I mean, the enzymatic reaction is taking place in all the ions, just mostly with Mn. So C is not very assuring. D. Mn2 plus increases enzyme activity to a greater extent. That's true as demonstrated by the error bars without, there's no overlap with the rest. Okay. Many species of birds protect themselves from predatory birds by emitting warning calls, then gathering together and chasing the predators. Researchers studied the responses by Paris major, a species of small seed-eating songbird, to two predators, tawny owls and sparrowhawks. The tawny owl is a low-threat predator to songbirds because the tawny owl prefers to eat small mammals, right? So tawny owl is not a big threat to the songbirds. That means that they won't even, spend too much time emitting warning call toward it, probably. Okay, now why? Because the tawny owl prefers to eat small mammals, maybe like, mice, let's say, and only occasionally eats small birds. But the sparrowhawk is a high threat predator. That means when they spot one, they must emit lots of warning signs or warning songs. Okay? Now, the researcher first determined the major characteristics of warning calls produced by the songbirds when they encountered lifelike models of tawny owl or sparrowhawk. So they did not use real predators, they just used lifelike model. Let's analyze the table. It seems that the length of the call, let's say the freak call, right, because they're freaked out, is higher for the sparrowhawk, and there is no overlap. The number of notes within a call is going to be higher for the sparrowhawk. The time between the notes in a call is going to be, again, higher in sparrowhawk. And the call rate is going to be also higher in sparrowhawk. In a second study, the researchers used speakers attached to trees to play recorded warning calls produced by the songbirds in response to tawny owls or sparrowhawks. The researchers performed several trials in which they analyzed the response of individual songbirds to the warning calls. For each trial, they determined the shortest length of time for the first songbird to fly within 6 meters of a speaker, and how close the bird flew to the speaker. The speaker, of course, resembles the what? The predatory bird, exactly. So, the shortest time for a songbird to approach the speaker is going to be much higher in the case of the sparrowhawk. And the shortest distance between the songbird and the speaker is going to be also much higher when it comes to sparrowhawk. So they're actually very terrified of the sparrowhawk. They approach it, get close to it to see if it's actually a sparrowhawk, then they're going to start to emit warning signs. Based on Table 1, which of the following best describes a change in the warning call of the songbirds as the threat level of a predator increases? Okay. As the threat level of the predator increases. So this has to do with Table 1. Okay. Let's bring it close to us. Okay, so here we have the table. It says that describes a change in the warning call of the songbird as the threat level of the predator increases. Okay, so who is a more of a threat to the songbirds? The tawny owl, or the sparrowhawk? Sparrowhawk. Right now we're focusing on the sparrowhawk. So it says in A that the calls become shorter in length. Actually, the length of the call becomes longer in the case of the sparrowhawk. So it's not A. The calls contain fewer notes. Actually, a number of the notes in the call become more in the case of the sparrowhawk. The calls have less time between notes. Actually, time between notes in the call is more in the case of the sparrowhawk. The call rate has increased. True, the call rate has increased. So it's going to be choice A. Good. Okay. Which of the following claims about the warning calls produced by the songbirds is best supported by data in Table 2? Now I'm going to go ahead and bring up Table 2 here. Okay. So which of the following claims about the warning calls produced by songbirds is best supported by data in Table 2? The different warning calls determine the frequency with which the parent songbirds will leave their nest to protect their young. Did you talk about the frequency by which they leave? No, it's not mentioned in Table 2. The different warning calls reflect how the songbirds approach each other to I don't know what that is. Chase predators. Okay, maybe. I mean, they have shortest time is different and shortest distance is different. Maybe that's why. The different warning calls determine the likelihood that a songbird will be attacked by the predator. Maybe. The different warning calls protect not only the species of songbirds, but also other birds. Now, we don't know that. We cannot tell that, right? Okay. Okay, so what I see is that the songbird, the time of the songbird to approach the speaker, which is the predator, and the distance of the songbird from the predator or the speaker is basically different if we are, comparing tawny owl to sparrowhawk. We have different songbird response. Okay, and so if there is a sparrowhawk, they simply will take less time to approach it, and they also approach it with lesser distance. That's going to be a different behavioral response, right? And yeah, so most likely B. But let me just demonstrate something else. If we go back to the beginning of the question, you will see how they said that many species of birds protect themselves from predatory birds like sparrowhawk by emitting warning calls, then gathering together and chasing the predator. So we should talk about that, chasing the predator somewhere here, right? Which is choice B. Okay? Good. Now, producing a bird call with multiple components is a complex process. There is a wide range of variation in the particular type of a call produced by the individuals in a population of the songbird species. Which of the following best explains the wide range of variation in any particular bird call? Okay, so if you have variation, you probably have, the interplay of multiple genes or the effect of the environment on the gene, right? So either multiple genes working together, or maybe the environment affects the gene expression. Two possible reasons for this variation. So A is okay, right? Not a single gene, no. I mean, if maybe a single gene with environment, but there's nothing about that. Linked gene, in fact, results in least variation possible. And yeah, so it's going to be choice A. Okay. At one time, farmers planted seeds treated with pesticides to protect the seeds from being eaten. Based on the information provided, which of the following most likely explains why this treatment greatly reduced the sparrowhawk population size, but had much less effect on the tawny owl population size? Question, what does the sparrowhawk eat? The sparrowhawk eats small birds, but the tawny owl eats small mammals. Okay, and so the small birds who are, um, let's see, based on that. Okay, so let's see. The seed-eating birds were a larger portion of the sparrowhawk's diet than of the tawny owl's diet, and the pesticides build up to toxic levels. Yeah, this is called biomagnification, which basically means that toxins, such as pesticides, build up as we go up the food chain, as we go up the trophic level pyramid. And if you're at the top of the pyramid, like a sparrowhawk, you will get most of the toxins. What happens is something like the eggs don't hatch in time, or the eggs basically break before the hatching time. And so that's going to be choice A. Okay. 8. An estimated population size of the songbird species in Europe is 130 million birds. If in 1 year, okay, there are 1,500 births and 1,200 deaths for every 1,000 births, which of the following is closest to the net growth of this European songbird population of the year? Alright, so what you will do is you will take the births minus the deaths per every 1,000 birds, and you will get you will get 300 over 1,000, which means 0.3. That's the growth rate. Okay, 0.3. They want the net growth. You will take that 0.3 and you multiply it by the population of 130 million, and you will get 39 million. Okay. Question 9. Which of the following statements describes a mutualistic relationship, which means a relationship where two organisms help each other out, between organisms of two separate species? One organism benefits at the expense of the other. That's called what? That's called parasitism. One organism obtains resources more efficiently than another organism. That's like more like competition. When the organisms are separated, neither survives as well. That's true. When they're grown together, they survive actually better. Okay. So here we have bar graph, and okay, comparison of the percent of species in different groups of organisms that are threatened with extinction. Okay. Researchers evaluated the numbers of native species in Australia over the past 200 years. They determined the percent of species in different groups of organisms at various threat levels of extinction. Based on figure 1, which two groups of organisms include the greatest percent of species that are not threatened? Not threatened, that means you have to have a gray bar, correct? A gray bar. Now, reptiles and fish. Actually, reptiles do have some level of being vulnerable, right? And they also have some level of being endangered. That's not so good. Reptiles and plants, even worse, right? So like if you do reptiles and fish, oh, by the way, the fish, some actually already went extinct. So A is really bad. Reptiles and plants, again, plants have some endangered and some vulnerable, but it's okay, we'll keep it on the table because there is no extinct at least. Okay. Invertebrates and fish. Invertebrates are not threatened. And fish, they have some extinct, but they're mostly not threatened. So actually, C is a very good option. Invertebrates and reptiles. Okay, right. So fish have already went somewhat extinct, correct? But the reptiles have not gone extinct yet. So they have a chance of being saved. So here, which two groups of organisms include the greatest percent of species that are not threatened? It must be the invertebrates and the reptiles. Okay. Actually, looking back, invertebrates and reptiles have not so much of a gray bar, right? Not so much. It starts here. But then if you look at invertebrates and fish, the fish start here. So it has to be the invertebrates and the fish. So it's C. Okay. So in here, we see that it talks about DNA composition for nuclear DNA. And they want to see if the composition of nuclear DNA rules for nuclear DNA are the same as those for double-strand mitochondrial DNA. Scientists determine the percentage of each nucleotide in a strand of double-strand mitochondrial DNA. Okay. Which of the following data sets best represents the expected data of the analysis? Now, so what do you expect when it comes to DNA base pairing? You expect to have an equal amount of A to T and also an equal amount of C to G, and the whole thing to add up to 100%. Right now, here we have A and T, and here we have G and C, and they are found in equal amounts, right? So G with C equal amount, A with T equal amount. So it's going to be choice A. That's what you expect, basically. Okay, 12. Scientists recently discovered that long-term memory formation may be partly controlled by stimulating protein synthesis with a protein called, I think that's ELF2 alpha, in a specific brain cell type. ELF2 alpha works at the initiation step of translation. Initiation means the beginning. Which of the following best explains how ELF2 alpha promotes protein synthesis? So, promotes protein synthesis. So, to make protein, you have to translate an mRNA, correct? Okay. Now, it says works at the initiation step of translation. So your focus right now is on translation. So is A translation or transcription if you're making RNA? That's transcription, right? We're not focused on transcription, we're focused on translation. ELF2 splices DNA together to make mRNA. Again, you're making mRNA, you're not making a protein in B. ELF2 binds to polypeptides and causes them to fold into proteins. That means we already made the protein, it's just folding it. That's not initiation. We're looking for the beginning, the initiation. Forms a complex with mRNA, which binds to the ribosome. And once the mRNA binds to the ribosome, you can now start initiation. You can now start the translation. So it has to be that. Okay. Question 13. Aminotransferases are enzymes that play an important role in nitrogen metabolism in all living organisms. An aminotransferase catalyzes a reaction in which the amino NH2 group is removed from the carbon backbone of amino acids. Okay. The amino group is a major source of nitrogen required for metabolic processes. The carbon backbone of many of the amino acids are incorporated into intermediates of the Krebs cycle pathway. Okay. The asterisk represents the intermediates that can be found from the carbon backbone of amino acid. Again, we are removing the amino group from the carbon backbone of the amino acid. So like this guy here has had the amino group removed. This guy here has the amino group removed. Same goes for the oxoacylate, for the fumarate, for the succinyl-CoA, and for the alpha-ketoglutarate. All of them had their amino group removed from the carbon backbone. Okay. A particular aminotransferase plays a role in the conversion of amino acid aspartate into oxaloacetate. So aspartate becomes oxaloacetate by the amino acid, by the enzyme aminotransferase. And this is going to be a component of the Krebs cycle. We have the reaction here involved. As you can see, the aminotransferase is able to remove the NH2, and that will convert the aspartate into oxaloacetate. And then it's able to give the NH2 to pyruvate so it can become alanine. Okay. Which of the following best describes the reactions of the Krebs cycle in eukaryotes? Of course, Krebs cycle occurs with the help of an enzyme. Okay, this is background knowledge, but yeah, it's just background knowledge that Krebs cycle requires enzymes. And I'm sure that it's also mentioned here. Let's see, our presentation. Right, so here it says a particular aminotransferase enzyme. So anything with ACE is going to be an enzyme. So this enzyme is going to be playing a role in the Krebs cycle, basically. So it's not A. The reactions are identical to the reactions of the Calvin cycle, but occur in the reverse order, very different. Calvin cycle occurs in, it's just a different pathway completely. Calvin cycle is building for you a glucose. The reactions occur in the matrix, that's true, right? The Krebs cycle occurs in the matrix of the mitochondria. Okay, based on the pathway shown in figure 1, which of the following best explains how the carbon atoms of amino acid backbones that became a part of the alpha-ketoglutarate might end up in citric acid? So let's just go back here and look for alpha-ketoglutarate. Okay, so right now our focus is going to be only on alpha-ketoglutarate. Okay. Okay. So alpha-glutarate can be broken down, transferring carbon to... We should just go back to the figure, okay? Let me bring it up here. Okay, so here is our figure. It says alpha-ketoglutarate can be broken down, transferring carbon to isocitric acid and then to citric acid. But alpha-ketoglutarate is here. Isocitric acid and citric acid are behind it. So it's not going to go backward. It will just keep going forward. So it's not true. Okay. Carbon atoms in the CO2 that is released during the conversion of the alpha-ketoglutarate to succinyl-CoA can be fixed into citric acid in the next round of the cycle. Again, we have to go backward to see that, which I doubt will work. Just keep going forward in your diagram. Carbon atoms that carry energy may be released from alpha-ketoglutarate and added to any molecule in the cycle, including citric acid. We have to go backwards, so maybe. Carbon atoms in alpha-ketoglutarate are found in subsequent molecules of the cycle, including citric acid, as one molecule is converted to the next. Let's see. This is what best explains. So it will be found in subsequent molecules. That is true. And citric acid is, so if you go all the way back, you at some point go back to citric acid. Okay. So that's going to be the best answer, that the carbon atoms in alpha-ketoglutarate are going to be found in subsequent molecules, like maybe as 6-phenyl-CoA, maybe as succinate, and so on, going all the way back to citric acid. The answer is going to be D. Okay. Now, based on figure 2, which of the following best describes the reaction catalyzed by the aminotransferase in the production of oxaloacetate? Yeah, so here's figure 2. In A, it says the aminotransferase removes an amino group from aspartate and transfers it to oxaloacetate, which is actually very true. This NH2, is, one second, removes an amino group from aspartate. It removes it. Actually, that's not true because it removes it, but it does not give it to oxaloacetate. It does not. So A is off. The amino transferase removes an amino group from aspartate. That's true. And gives it all the way to pyruvate. Yeah, true. It will give it to pyruvate. And that will make pyruvate into alanine. So B is the right answer. Let's see why the others are wrong. The aminotransferase removes an amino group from alanine. No. The aminotransferase removes an amino group from alanine. No. Again, no. Good. Okay. 16. Based on figure 1 and 2, which of the following changes to the Krebs cycle is most likely to occur if the aminotransferase that converts aspartate to oxaloacetate is non-functional? So if you don't convert aspartate to oxaloacetate, what is gonna happen? Okay. So let's go back to this figure 1 and see what happens with oxaloacetate. So if we look back at figure 1, which is this one, let me just copy it. Okay, so if you don't get to make oxaloacetate, then this step will be stopped. Okay. And if this step is stopped, are you going to be able to make citric acid like before? No. Okay. So it's going to be choice A. And of course, if you don't make citric acid, you cannot make NADH, which will be made in 3 subsequent steps. So it's going to be choice A. Okay, an experiment designed to answer which of the following questions is most likely to help scientists determine whether a newly identified gene codes for the aminotransferase enzyme. Okay. Is the gene located near genes of other aminotransferases? The location of the gene does not matter. It's about does the gene get to code for that specific enzyme or not. The location does not really matter. Is the gene expressed in all cells of the organism in which it was identified? No. Not all the cells will express the gene. Only the cells that require this enzyme will express the gene. That's not true as well. Is the nucleotide sequence of the gene similar to that? Okay, so is the nucleotide sequence of the gene similar to that of an aminotransferase in a closely related organism? That's probably going to be the one. Because if we have the same sequence in a closely related species, then you get the same type of an enzyme from the expression of this gene. Okay, so this is called homologous structure or homology, basically. That's the best way to know if the newly identified gene codes for the same enzyme. Okay, question 18. We have two types of immune system cells, macrophages and betas and B cells, and also red blood cells have different functions and different structures. Which of the following best explains the relationship between the size of cells shown in figure 1 and the cell's ability to function? Now, as we know, the smaller the cell, the better, right? The smaller the cell, which is the case for B cell and red blood, actually no. So red blood cell is going to be the smallest cell. It has the highest surface area to volume ratio. Do you know this? You should know this, right? Just look at the size being the least diameter. And so they're going to be the most efficient at doing their job. 19. Which of the following best explains mechanisms by which sexual reproduction increases genetic variation? Of course, you're going to have things like meiosis. You're going to have things like random fertilization. And look for stuff like that. So what's special about meiosis? Crossing over and also the independent assortment of the chromosomes. And so it's going to be choice C. Good. Okay. So here we have the effect of exercise on the concentration of LAC-PHE. And we're measuring the time at which LAC-PHE was measured before exercise, immediately after exercise, after it by 30 minutes and after it by 60 minutes. Researchers studying the effect of exercise on the signaling molecule LAC-PHE measured the LAC-PHE concentration in the blood of resting mice as well as in mice at various times after the exercise. So based on the table 1, which of the following best describes the relationship between exercise and the concentration of the LAC-PHE? Let's go back to the table. So it seems that before the exercise, the amount of the LAC-PHE and 60 minutes after the exercise, they are, let's see if they're overlapping or not. So here we have 0.21 to 0.29. And here we have 1.2 to 1.35. And here we have 0.57 to 1.67. And here we have 0.19 to 0.23. Now, do we see any overlap? Yeah, we do. Before the exercise and after the exercise by 60 minutes, we see an overlap. Okay. But it's highest immediately after the exercise. That's when it's going to be, it's at highest levels, basically. Okay. All right, so the time required after exercise for the concentration of LAC-PHE to return to before exercise level is 30 minutes. Let's see, the time required after exercise to return to the, actually no, it required how long to return to before the exercise? It required exactly 60 minutes, right? Because after 60 minutes, their levels start to overlap again. So it's not 30 minutes, it's actually going to be 60 minutes. The longer the time after the exercise, the higher the concentration of LAC. In fact, no, because after 60 minutes, it's at its lowest. So no. The concentration of the LAC-PHE is highest when the mice are at rest. No, at rest means before exercise. It's not highest before exercise, actually highest after 30 minutes. The concentration of the LAC-PHE is highest immediately after the exercise and then decreases. Yeah, it's actually highest immediately after the exercise, right? So it's going to be choice D.